Alright, AP guys, I'll go ahead and get started. We're going to cover chapter 15. Obviously, we just got done with Mendelian genetics in chapter 14. Our test over this unit 14 and 15 combined together is going to be, you got it, next Valentine's Day, February 14th. So next Thursday will be our test over 15 and chapter 14. So some Mendelian genetics. Happy Valentine's Day. All right. Basically, 15.1 opens up with a little bit of a basic background of Mendelian inheritance and how it affects the chromosomes. You can read into that. I'm not going to cover that too much. Here's some basic things. If you need to fill in your notes, they can come from this slide here. The other really important thing is the chromosome theory of inheritance. And basically, the chromosome theory of inheritance states two things. Number one, the factors that Mendel mentioned, the genes in other words, have a specific location, on the chromosome. We call that, and like I said, that's just really the position where it's found. The second major thing that the chromosome theory of inheritance states is that the chromosomes undergo segregation and independent assortment. Once again, both these two things, not too revolutionary, but they're the keys to kind of understanding gene mapping and what we're going to talk about upcoming. You look at this diagram here from your book, basically kind of shows it as we need to see it. When you look at two traits for any particular trait, we have this fertilization occur from the P1 generation, and then here are the two different very important laws. Law of segregation on the left side, law of independent assortment on the right. When you see that, law of independent assortment is really talking about these particular traits. Independent assortment is talking about this part right here. What are we going to actually end up getting? Where they actually end up dividing out. Amongst that gets us the different offspring. So what we're going to focus in on is looking at how do chromosomes play a role in this whole process. So the first solid evidence for associating the specific gene with a chromosome came from a guy by the name of Thomas Hunt Morgan. And Morgan was an embryologist, studied obviously the embryos of different species, which at the time, since about Darwin on, this was a really big thing to do. It's pretty easy to do. With lizards and birds, you could easily go find them in the wild at different stages or different time periods and be able to detect the different stages of embryonic development. And one of the things that Morgan was really interested in was fruit flies. And basically what fruit flies did for him were they provided a convincing evidence that the chromosomes were basically the location of Mendel's factors. So we get this Mendelian factors are going to be found on the chromosomes. Now obviously when we talk about that, we're talking about alleles. That's kind of Darwin's word. We're talking about factors. In other words, we're talking about genes. Genes are found on the chromosome. And it wasn't until Thomas Hunt Morgan that we really understood that. But the most important thing you got to remember about Thomas Hunt Morgan was what he was a little different than other people at the time. And it was because of the species that he worked with, because of fruit flies. This is really what's going to help him kind of put himself on the map, pun intended. The fruit flies are going to be an important species to study. It would be a great SAT question doing something along the lines of Mendel is equal to peas as Morgan is equal to. So, why did Morgan choose fruit flies? It's not that they're cute and cuddly or anything like that. It'd be a lot easier to, cooler, I think, in my own opinion, to work with something more badass like a, lion or tiger or something like that. But there's three things basically that Morgan really liked about fruit flies and made them a good study. So the first thing is they were able to breed at a very high rate. You can look in these little tubes down here at the very bottom of the thing. All those little dots are fruit flies. You can have 200 or so easily in one of those little units. They're easy to keep. They're easy to reproduce. They don't take a lot of food down here. That's their food supply. So it's just a mixture of some carbohydrate, sugar. Get their water. You put a little styrofoam cup in there. Put some webbing for them to lay their eggs, and that's about it. You let them go. don't need sunlight, don't need water, don't need to take the cap off. They're good to go. So the first thing is they're bred at high rate. Second thing is the generations can be bred about every 2 weeks. You'd get a generational time. So, in a course of a couple months, you could have 8 or 10 generations going on really easily and really see evolution working. Number one, that's why we use them for evolution studies today. But they're also really good for genetic studies as well, seeing how traits pass on. The last reason that they're really good is they only have 4 chromosomes. Having those 4 chromosomes is going to be really important because, number one, it's easier to find exactly the location. Any gene for humans, for example, could be on any one of the 23 sets of chromosomes, and figuring out exactly which one it is can be extremely difficult. Not a problem when you only have 4. You got a 25% chance of it happening on any chromosome. Another trait, fruit flies, as simple as they may seem, have a lot of different characteristics that can be easily identified underneath the microscope. Wide-eyed versus red-eyed. You could have the wing shape. Look at the spots on the wing. Spots on the wing versus no spots. Okay. Look at the body, the abdomen. Legs. See this little right here, this leg right there? Let me put a little line onto it. Right there. Point to it. Notice how here we have an actual full leg, we have a little stubby leg here. Big difference. Okay, same there. These spots versus those spots up here. Eye colors, stripes on the back, bristles of the hair, all lines on the abdomen, all easily identified traits. Wings also, vestigial versus non-real wings. And therefore, they're easy to see the traits underneath the microscope. Here's some different types of examples that we see. Just for the, if you look at the head, the antenna right here, bristled versus short, gray versus black, red-eyed versus a cinnabar, which is just a little bit darker color, normal wings for those vestigial wings, brown-eyed, red-eyed, and they all have different locations on a particular chromosome. Okay. When we talk about this word right here, we'll come back to the wild type versus mutant type. When you look at any trait, these two terms are going to be important for us to look at. So one of the things that Morgan noted when he did this were that phenotypes, there were certain types that were normal in the common fly population. And he called those normal types the wild type. Now, it does not mean dominant, it does not mean recessive, it just means the normal. So that's a big difference. A lot of people want to associate this with, the concept of being a dominant trait. We can't do that. Okay? Traits alternate. The ones that are not the dominant type are called the mutant phenotype. So red-eye is the most common, wide-eyed is the mutant phenotype. So, why does this matter? What is Morgan kind of famous for? So one of the things he did is he made it male flies with white eyes that were mutant to female flies with red eyes that were wild type. Now when you do this, if you expect it just a normal, if this is, the red eyes is the dominant characteristic and the white eyes is the recessive, you'd expect a ratio that all of the F1, they all should be red-eyed. And that's exactly what he saw. Okay, so no shocker there. When you look at the F2 generation, you should expect when you cross 2 of these to get a 3-to-1 ratio of red to white. Once again, that's exactly what he saw. So it's just fitting Mendelian genetics to a T. When he crossed and got, but when he really examined this, when he looked at this particular characteristic for the 3-to-1 ratio, he noticed something different, okay? That in that 3-to-1 ratio was actually hiding because only the males, only the males had the white phenotype. So of this one, they were all male. Now, that was something that was kind of surprising to him, okay? Only males had white eyes. There's no females that had the white eyes. So what Morgan determined was that wide-eyed mutation allele must be located on the X chromosome, on the X. And once again, this evidence supported the chromosome theory of inheritance, and that genes, specifically certain genes found only in male populations, would be found on a sex chromosome because they did not have the complementary pair like a female does. When you look, once again, this is what's expected, all red-eyed, but the problem is we did have only males had the white eyes. Basically explaining what happened, since there's no gene that counteract the lowercase w here for white-eyed, when this occurred, you had white-eyed males that were the condition, no females that had the condition, and some males that also had red eyes. So that's Morgan's one of his major contributions, is sex-determining genes, that the X chromosome was not only there to determine the sex of a species, which that's what it was. There was also some traits on there. So this is the important part. This is what you kind of want to focus in on, how to do some gene mappings and where this all plays in. This is what the rest of the class, when you're done with this video, is going to be about. Linked genes tend to be inherited together because they're located near each other on the same chromosome. Okay, and when you look at any particular trait, gene, you look at the fact that each chromosome has hundreds or thousands of genes on there. Okay, and then the genes themselves are always going to be located in the same location on the chromosome. Genes that are found similar or near each other are going to be linked together. They're more likely to be passed on. Because when you have crossing over, this whole chromosome is going to go, or if there's crossing over event, a gene that's found here is more likely to be passed on with a gene found here. It's not a guarantee, but it's more likely. Whereas a gene here and a gene here are less likely to be passed together because of crossing over. If this were to cross over up here, but not down there, you pass on the A for the one chromosome, but the B's would not. So even though they're found on the same chromosome, we wouldn't see them both being passed on. Okay? And what he did was he noticed that there were certain genes that were being linked together while others were not. And he crossed, his kind of famous experiment was when he crossed the body color and the wing size. And he looked at those two traits. Now, once again, a couple things you got to remember. A lot of generations, a lot of offspring he can get data on. And the most important thing, there are only 4 chromosomes to select from. So you got a 25% chance of any two traits being on that same chromosome. Okay. So here's his cross. He took what we call the wild type. Once again, it doesn't mean dominant necessarily, but it means the most common condition. And it was a gray-bodied normal wing. So if you looked at normal fruit flies, this is what they would have looked like. He crossed that with a black-bodied vestigial wing fruit fly. Okay, that was his original parent, and it got this hybrid species right there. He then did a test cross, because we know what a test cross, we're going to cross it back to something that we know. We know the genotypes of that just off of because it's completely recessive. Okay. Now, when he did this test cross, what he found was that some of the population looked just like one of the parents. So this one looked just like that. Okay. And that this one looked just like that. But there were 2 that were a combination. They were recombinant. They were not like either one of the parents. Okay. Something different was going on here. If they were linked together, all of the population should look like these 2. But that's not what he saw. He saw something going on here with about recombination. So we call this the recombination frequency, and we calculated that in labs. Calculated that in one of our previous labs. So from these results, what did he do? Morgan reasoned the body color and wing size are usually inherited together. The key word there is usually inherited together in a very specific combination. Okay, because the genes are on the same chromosome. Because they're passed on, and there's that amount of parental genes, they were linked together. In my best Stephen A. Smith, however, non-parental phenotypes were produced. The parental phenotypes would be expected or explainable, or even if you got all non-parental phenotypes, you could explain that. But the fact that there were non-parental along with the parental phenotypes That's what's going to be important for Morgan to kind of understand or comprehend. That, yeah, there were parental, so therefore the genes are linked, but they should be 100%. All should be one of the parent or the other. It's not what he saw. Most of the offspring, okay, when we looked at it, were like the parents, but not all of them were. So what he had to figure out was exactly what was going on, and that's where we look at genetic recombination linkage. So how does this all work? We're going to take this information from Mendel, we're going to take what we know from Morgan, and we're going to actually do some genetic linkages and understand exactly how the genes are going to be found, okay? The key is looking at the unlinked genes, okay? Seeing exactly how this occurs. Because Mendel himself observed that combination of traits in some offspring differ from either parent. This is not a shock. Offspring with a phenotype matching one of the parental phenotypes, we call those the parental phenotypes. So the first thing you do whenever you do any type of recombination problem is you want to find the parental phenotypes, the ones that look like either parent. Doesn't matter if it's the wild type or if the double mutant type, you got to find the parental phenotypes, and you're going to count those up. Okay. If you have, then you're going to look for offspring with the non-parental phenotypes. It's a new combination of trait. Okay. We call those the recombinant types or recombinations. And you're going to get a total number counted there. 50% frequency of recombination is observed for any two genes on different chromosomes. So the first thing, whenever you look at these, if you get a 50% recombination between two traits, then you know they're just found on different chromosomes. If that number is not 50%, then something else is going on. Okay, and that means that they're found on the same chromosome. And if they're found on the same chromosome, we should be able to find the distance between them on a particular chromosome. First thing, look at the percent recombination. If it's 50%, then you know they're found on different chromosomes. You take a look here, take a look at the cross that we have. Parental types, offspring are found here. The recombination offspring are found over here. When you do this, based on the ratio, 50-50 is what we should expect to find in our numbers. So, one of the things that he finds, obviously they're incomplete. Morgan proposed that some process must sometimes break the connection between the genes in the same chromosome. Now, we talked about this process already in the previous chapter, and that mechanism is crossing over. Okay, so he makes a prediction about crossing over before it's actually discovered. Crossing over between two chromosomes, I know some of you, one of the Davises at least, will be probably making fun of me for this diagram. There my tetrad. Okay, but that's where the gene is, a gene that's found here, and here they didn't cross over together. Okay, so therefore they're not going to be the same. When you take a look at the actual combinations, you notice here there's something going on. Recombination frequency. There's a total number of offspring of 2,300. What we want to figure out is the recombination. Typically, if they're found on different genes, it's going to be somewhere very close to 50%. Okay. Now in this case, we don't see a 50% recombination. The numbers are here and here, much larger than these recombinations. Parental types are always the highest percentage. So the first thing you do whenever you look at this, because we're going to have more than two traits, we're going to have 3, 4, 5 traits on a particular chromosome. First thing you do is you look for the highest number. Those two things will always be your parental types. Then we look at the second numbers, which are going to be your recombinations. Okay, they're always a little bit lower. And as long as these two numbers add up, do not equal 50% recombination, then you know that those two genes are found on the same chromosome and they were not passed completely together every single time. Sometimes they were. And when they were, we got one of these two parents. But when they weren't, we got one of these two parents. Okay, so it's going to come up to another person. It's not going to be Morgan who kind of figures out the really cool part of this. So I'm going to tell you a little background about one of his students. Good old Alfred was a college student at Columbia University, and Morgan was a teacher. He's kind of revolutionary at the time in the area of genetics. Everybody kind of likes him. He's kind of the hot guy in genetics. So just imagine, I always think of some people today that are big in kind of the scientific community. If you decided to go work for one of those guys to do research. Also, his teaching techniques, I guess he was really engaging. The students liked him. It wasn't just that he sat there and lectured. Really big in experimentation and work. So two of his students, and one of them is Alfred, in his class, soon as class is over, he's a freshman in college. He runs up to Morgan and says, I want to work with you. So Morgan gets him a desk in his fly lab, which is basically a modified room which has just tons and tons of fruit flies, posters up on the wall, fruit flies all the time. Alfred's hooked. He's there nonstop. He basically lives in this room with Morgan. And it was a really good experience for both of them. They kind of talk, and they talk about different research, and hey, I'm gonna research this, I'm gonna cross this with this. And it was really kind of a time of pioneering for both of them. Okay. Now remember, Morgan's is the professor. He's supposed to be the lead. And what happened is Alfred one night was just kind of talking to himself about recombination, and he had this idea popped in his mind that the further away two genes were, the higher probability that crossing over will occur between them. And therefore, the higher the recombination frequency. Let me type that out for you. So that was his big breakthrough. Sorry for the typo. Obviously, there needs to be an I right in there. So this is Alfred's big breakthrough. And once again, it's kind of interesting. The side note on this is kind of cool. Maybe you can get away with this in college if you ever don't do a homework assignment. He basically just blew off all his undergrad work, blew off any homework that he actually had, even for Morgan, just to work on this one theory. And what was really cool is it was a pretty accurate way to do it. He figured out how to map out the genes on a chromosome. This is a revolutionary idea. We can now, based on how they recombinate, figure out distances. A couple key points, though, that we need to make about this. It is really cool, and it shows how far apart they are, but it doesn't tell us everything that we need to know. A linkage map, okay, does not tell us the exact location. It does tell us distances between genes. And we represent those distances on a gene as a map unit. Okay, so the one thing that we talk about is a map unit. The more common phrase that Alfred named it after his mentor is a centimorgan. Same thing, okay? And both these two numbers equal 1% recombination. So if a trait is 17% recombination, therefore it is 17 map units or 17 morgans away. Map units do not tell us the exact location. It tells us the order, not the precise place. But if we can find out where one gene is, and we know how far away another gene is from that particular gene, now we're cooking with gasoline. Okay. All right. What I want you to do now is go to that link I have for you on the bottom of the page, library.thinkquest.org/20465, blah, 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 blah. And on the blue sheet, I want you to kind of follow along and fill out that blue sheet off of that website. So you should have got a packet. Front sheet should be a blue sheet. Okay? Go ahead and do that now. Pause this video, put me on the background, listen to your music. You probably listen to some thrift shop or whatever the kids on the street are listening to today. You know, whatever you guys find as music. Awesome. Not really. All right, welcome back. So just as you review, notice that when we look at a particular trait, we have a map distance between two traits, 17%. In this case, we also have percent distances for the other two. One thing that I hope you realize, it's not exact. 9%, 9.5%. I know you guys aren't, all super geniuses at math, but everybody should realize that it doesn't equal 17%. Okay, there's a lot of factors that kind of go into that. These are relative distances from each other, relative locations. That's fine. Biggest problem that people have when they work on some map distances, they want them to work out perfectly. It doesn't work that way, okay? Now, every once in a while, we'll give you a problem that it does just to be nice, but typically, those numbers will not equal. Okay, we can make these maps of fruit flies using once we can figure out a location of it, okay, we can then go through and look at exact locations from one gene, find another gene. Okay. All right, last slide. Here's a particular trait that we looked at. Bunch of different characteristics. You can find the map distances between any particular trait. And as you can see here, some genes are closer than others. The exact distances are not known, but once again, it gives us a great starting point to exactly find where they are. What we're going to have you do now is I have the next sheet, which should be a green sheet, is going to be kind of explanation on how to go about actually figuring out some more map distances. It's going to be kind of a tutorial for you to follow on figuring out distances of genes on a map. Thanks. Hope you enjoy the video. I'm enjoying KC, I hope. And I'll see you guys on Tuesday where we'll kind of pick up 15.3, talking about sex-linked genes and how they can be seen in the population. Thanks. Have a good weekend. And the last sheet in your packet will be your homework. And your homework will be a white sheet. And there might be 2 white sheets just depending on if I get it done or not. All right. Talk to you later. Bye.